Hello, everyone. Thank you so much for coming to our tutorial session. My name is Mikaya Mayalu. I'm the chair of this session, along with my colleague Anya Bateka. The title of this session is Control Processes of Engineered Living Systems. And with that, I will begin as a first speaker. So today, I will talk about how we can control populations of cells using molecules that they secrete as a feedback signal. And also, I'll talk about some control strategies possible with this idea and what dynamics can result from this. Since this is a tutorial session, I will want to make sure that everybody is on the same level, so I will begin with some introduction for the topic. To begin with, I will give a kind of a definition of what synthetic biology is. So synthetic biology basically is the redesign of a biological system, often biological cells, in order for them to perform new tasks and that they potentially didn't have the ability to do before. So the redesign of cells is achieved through modifying genes within a cell or building them from scratch. And since cells are the instructions, or excuse me, since genes are the instructions that cells use to function, modifying them or adding new ones can radically change the cell's behavior. So multiple genes and their products can also be made to interact into and form networks. And these are also referred to as gene circuits. And these circuits are somewhat analogous to electrical circuits in that they can be designed and manipulated to control cellular behavior in a more predictable way. So the applications of these redesigned cells, termed synthetic biology, are vast. So in human health, we can think about using synthetic biology to design vaccines, gene therapies, cell therapies, and probiotic systems. Also in agriculture, we can use synthetic biology to design disease-resistant plants and drought-resistant plants, and also sustainable plants. There are also applications in industry and the environment, including bioenergy and bioremediation. And with these many applications, one main concern is how these cells can be redesigned, how these redesigned cells can be redesigned to be trustworthy, predictable, and robust. So that is a main concern with these cells. And so we can use control theory as a tool to design these circuits and also analyze the trade-offs between circuit design and especially designs that potentially produce similar functions. So furthermore, we can use control theory to design feedback loops at the single-cell level and at the multi-cell level. So the single-cell level, we can have basically a loop within the network inside this gene circuit. At the multi-cell level, we can have basically chemical signals which are released into the environment. And then the aggregate signal that is released by each cell can then be sensed as an input by the cell, which creates the feedback loop. And so these feedback loops can be used to increase things like safety, reliability, and controllability, which are the main issues when you run into designing these synthetic biology cells. And this is very helpful, especially with uncertainty in an uncertain environments. So actually, for the rest of the talk, I will focus on feedback loops at the multi-cell level. So these are loops that can, the cells secrete a specific chemical, and that chemical is aggregated in the environment, and then the concentration of that chemical is read as an input to each cell. And I'm specifically going to talk about this feedback in the context of cell population control. So feedback control of cell population is important for many applications, specifically therapeutic applications. We can think of if we are redesigning cells and inputting them into the human body, we want to make sure those cells don't invade or overgrow into healthy tissue. So having some type of control or feedback control over those engineered cells is a big issue and a big point. So cell populations basically need to be maintained at a constant level. And there's been some past research with bacterial cells that show this, to show how you can control density of a cell population, and this is through interfacing the signaling molecule, which has been used for a feedback signal, with a gene that causes death, which causes death of the cell. So basically, the signaling molecule acts like a feedback input to suppress the growth of the cell population. And this is referred to as negative feedback population control. So if we look at the null clients, the dynamics of this negative feedback population controller, if we were to plot the growth as a function of the feedback signal, the growth rate would be higher and then decrease as the feedback signal increases until it reaches the point of homeostasis, or the equilibrium point. And this equilibrium point will be stable, since above that point we have negative net growth, and so the cells will die until they reach the stable point. And then below that point, we have positive net growth, and so the cells will proliferate until they reach that stable point there. So this is a monostable system. Another architecture that could be of some interest is called the paradoxical feedback architecture. So this is where we utilize both positive and negative feedback in different signaling regimes. So paradoxical, the signaling works kind of like this. It's made up of two parts. So first, the cells produce a signal which is proportional to the cell population size. So they release that signal into the environment. And then those same cells detect that signal, and they can respond by either proliferating or dying. And that response is based on the concentration, the intensity of the signal that it detects. So if the signal is low, the detected cells respond by proliferating. And if the signal is high, the cells that detect the signal respond by dying. So we basically have the same cells, which again produce either proliferation or death, negative or positive or negative feedback in different signaling regimes. So it turns out that using this type of feedback architecture can cause the system to be bistable. So that is where we have the two stable steady equilibrium points with the unstable in the center. So an advantage of having bistable population dynamics for the paradoxical case is that it can be more robust to mutations in sensing. So, kind of a quick thought experiment. Let's say that we had this mutation where in the negative feedback case, cells could no longer sense anything. So before, they were sensing a signal, and that signal was activating mechanisms to allow them to die. So in the case where they can no longer feel that or sense that at all, cells will be in this no signal detected regime or low signal detected regime, and they will keep on proliferating. So in this case, these types of mutations will, if this was deployed in the body, would invade the healthy tissue, let's say. So let's think about the same scenario in the case of paradoxical signaling. So in the paradoxical signaling, if we had the same idea, let's say that we had kind of loss of connection of the cell with the signal. In this case, we're in a regime of negative net growth due to the bistability of the system. So instead of those mutated cells increasing without bounds, they would actually die out. So this is an advantage to this system, that in some situations of mutations, you could have this robustness to overgrowth. So another advantage to bistable population dynamics of the paradoxical system is that there is the opportunity for the system to switch between low-level population and high-level population, because there's two equilibrium points. So this can be achieved kind of by giving the system a perturbation. So the idea from my student here is that we have a signal, a similar signal, an extra signal that gets added to the environment. And that signal is similar enough that it perturbs the feedback signal into a different regime, the regime that allows it to, if the population started in the low equilibrium state, allows the population to expand, proliferate, until it reaches the high equilibrium point. So in this way, we can kind of transition between the two equilibrium points. And also, upon removal of the extra external signal, the population could revert back to contract back into the low steady state, or the low equilibrium point. So we wanted to test this out for a specific circuit, a specific circuit design in E. coli. So here, this is the implementation. So first thing we did was we had the external signal actually be an infection signal. So this is an infection that we might want to detect, let's say, within the human body. And so the cells will respond to this signal, kind of growing from the lower level population and proliferating to the higher level population. And then we also added an off switch. So this is basically a way for us to create the safety mechanism within our system, such that when the off switch is present, again, kind of a different external signal, the whole system would shut down and all the cells would be removed. And as well, now to implement the paradoxical feedback, we had to both have this negative feedback mechanism and the positive feedback mechanism. So to implement negative feedback, what we had the cell do was upon detection of the feedback signal, so a low feedback, or a high feedback signal, in the negative feedback case, the cell would produce a toxin. So this toxin would cause the cells to contract, basically die off. And then in the case where they feel a low amount of feedback signal, the cells would proliferate. And the specific mechanism we have here is drug resistance. So they would produce a drug resistance when they detected the low feedback signal. And the drug resistance would save the cells from dying due to the drugs which are in the environment. So those are the negative feedback implementations for this specific circuit. And lastly is the feedback signal itself. So this we use a very small molecule called the quorum sensing molecule. And it's basically being produced in proportion to the amount of cells present. And that is kind of the way that we can switch between the negative and positive feedbacks. And just quickly on the model, we can basically complete the model or implement the model with the components mentioned on the previous slide. And this is the kind of a set of differential equations which describe the rate of change of the signal concentration, or the feedback signal concentration, the rate of change, and which is coupled to the rate of change of the cell population. And the rate of change of the actual infection signal is also modeled here. And so the infection signal can influence the concentration of the feedback signal, or the total concentration, and that's what allows basically the population behavior to be perturbed. So this is a reduced order model. We assumed that the internal mechanisms that I spoke about earlier, which is the production of the toxin and the production of the drug resistance, are at steady state. So those are now parameters which are within the cell population dynamic differential equation. So this is a reasonable assumption because it's given the time scales of our specific system, the assumption that the internal dynamics have reached steady state makes sense. So the simulation results for this circuit reveal bistable dynamics, as we had previously proposed. And that is in a specific regime. So if we've maybe tuned the parameters of our circuit, we can tune different parameters within the circuit to affect positive and negative feedback. And we see that, depending on the strengths of each feedback, we can tune the system to be in the bistable regime. So within that regime, we're able to simulate this idea of transitioning from the low population to the high-level population. So in the first plot here, we see that the cells, there's no infection signal present, and so the cells will converge to the low population level. And then in the middle plot, we see that the infection is introduced, and so the cells respond by expanding, and they go to the high population level. Then when the infection is removed, the cells revert back, and they converge back to the lower level population. We could also test out the mutational robustness that we spoke of earlier, which is kind of eliminating the ability for the cells to sense any signal, and showing that in the case where we have some mutant cells with that ability eliminated, we're able to recover. So the mutant cells do die off, and we're able to recover the control of the cell population. Lastly, I want to briefly describe how the dynamics for the off switch, or the elimination of, complete elimination or removal of the cells occurs. So this is done by shutting off the connection for the cells to sense, basically shutting off the sensing connection. And for our specific configuration, if we shut off the ability for the cells to sense any signal, we'll have that the cells won't be able to produce drug resistance or any toxin. So in the absence of drug resistance, since there are antibiotics or drugs present in the environment, those will in fact kill off the cells. And there will be nothing to stop them from dying out completely. And so that's the main idea of how the off switch, which is irreversible, is implemented. So here is kind of the simulation of that. So we show that initially there's no infection. As the infection is introduced, the cells expand, and then even with the infection still present. When the off signal is introduced, our external off signal is introduced, the cells will die off completely. Lastly, some efforts have been put into kind of analyzing and tuning the sensitivity of this system. So it turns out that as you increase the sensitivity, if you want the cells to respond to lower levels of infection signal, you put yourself vulnerable to any disturbances. So if there's any slight disturbances that might erroneously activate or the expansion of the cells when you would prefer not to. So currently and this is the simulation showing how that could potentially happen, given the sensitivity of the system or how the parameters are tuned. So the main idea now is to optimize the threshold, the threshold for detection, while keeping in mind the threshold for disturbance rejection to get the optimal use case within our system. And then my student has also been looking at other approaches using agent-based models that allows us to simulate kind of a fuller system. So here we're showing how the engineered cells respond to the infection. And then we've actually added a treatment. So we've added a treatment module here where the cells then actually treat the infection. And when the infection is removed, the cells kind of contract back. And we've also shown here how the off signal can kill the cells, regardless of if the infection is present or not. So to conclude, we've basically shown how paradoxical feedback at the multi-cell level can exhibit these kind of bistable population dynamics. This bistability is useful for mutational robustness, as well as kind of this population level switch. And we've presented a specific circuit design in E. coli, but this methodology could potentially be used in different contexts and with different components. And the type of feedback also exists in natural systems, specifically in the pancreas with beta cells, which secrete glucose, and the glucose can kind of either positively or negatively affect the beta cells given their actual concentration. And so we're also currently implementing our designs with experiments and extending the theoretical work we've already done by adding treatment modules. And so lastly, I just want to thank my group and acknowledge my collaborators for this research. And I'd love to take any questions.
One quick question. I have missed out the information why the cell population would die out when the signal is 0. So you have some signal which is when it's 0, then the cells die out. When it's somewhere in between,
yes,
they proliferate.
Yeah. So you're asking why wouldn't the cell population die out when there's no infection signal? Basically, yeah. Basically, what? Well, so why do you have a non-zero low equilibrium point? So yeah, that's a question that I didn't touch on. So this system is supposedly theoretically going to be implemented within the gut, and it turns out that the gut has a natural basal level of the signal of the feedback signal. So in the basal level, the cells are, they're somewhat sensing that level, and they're not exactly at 0. So the basal is some offset from 0. Yeah. Is there any more questions? Yes.
How do you see this field develop? It's biologists and doctors and engineers and mathematicians. How do you think it's going to develop in the future?
Well, hopefully, as you said, we can get bioengineers involved and also, people from the medical school involved. I've been trying to collaborate with, gut biologists and, also, bioengineers and mathematicians and things like that to try to get this started. And yeah, I hopefully this will be kind of something that we can use kind of in parallel with clinicians to kind of, see what they need and help them, with their.
How did you get into the field yourself?
Oh, well, I'm actually a mechanical engineer. I trained under a robotics lab, to be honest. And then I've always been interested in kind of therapeutics and medicine. So during my postdoc, I kind of explored this research. Yeah.
Good career for an engineer.
Oh, yeah. Thank you.
So since this is kind of a tutorial session that is meant maybe also for students that might be interested in the field, I will start very slowly and talk a little bit about what our field does. So the title of my talk is The Rapid Development of Modular Synthetic Biological Circuits Using Designer Proteins. I'd like to first give a really brief introduction to what systems and synthetic biology are. They're great places where we can apply our control theory knowledge as control theorists. So since synthetic biology has had a lot of applications that are related to biotechnology, industrial and environmental biotechnology, it's shown its application in health and medicine. There's really a lot of areas that it's relevant to, and we can use our engineering knowledge to make it better, I believe. And then systems biology has held the promise to things like better cancer treatments and healthy aging and making better microbial therapies. So what is our role as engineers in this field? So I think the way that I think about synthetic and systems biology is that Systems biology is more engineering-y in that I don't need to understand every single component or model every single component of the laptop that is in front of me in order to get it to work. It's kind of based in the idea of like emergent properties, which is what we have in physics when we think about gas in a balloon, hot air balloon. Whereas synthetic biology is more like we are trying to build something from bottom up, and we are trying to figure out how to do that without knowing all the details. So here in synthetic biology, we've introduced already, Mikael has introduced already the concept of a circuit, but I wanted to go a little bit through it. It is the case that synthetic biology has relied a lot on the principles of electrical engineering. So instead of having things like capacitors and transistors and whatnot, in biology we often have like genes. And one way that they can interact with each other is that they repress each other's production. So if you have a lot of one, you have a little bit of the other one. So let's just follow along how this little circuit here works. It has 3 genes, A, B, and C. If you have a lot of A, you will have very little of B, and then you'll have a lot of C. And then it will give you a little bit of A, and then a lot of B. And you can hopefully see how it oscillates, right? And then on the right-hand side, it's a very similar version of it, except it has an extra gene. So if you have a lot of A, you get a little bit of B, a lot of C, a little bit of D, and you don't have to oscillate anymore. So this is very interesting. If you change the system even a little bit, can the models that we usually make in engineering be predictive of how the biological system that you have changed works? And I think that predictive models are key to understanding and engineering these synthetic circuits that are made up of many components and increasing complexity. Just to draw, again, a comparison between systems and synthetic biology perspectives, the way I think about it is that in systems biology, we try to do analysis through predicted models of abstracted layers below. So we don't need to know all of the details of every single biomolecule or gene to be able to tell like what an organ does, because we've abstracted that away. Whereas in synthetic biology, we try to perform design through modularity and robustness to the things that we don't know in the layers below. So then, my broad question for the talk is, how can we design systems that are robust with respect to this lack of information in the layers below? As you will see, one of the central themes of my talk is that you can't measure everything in biology. You can't have a perfect understanding of how every single thing works. And you shouldn't have to. You should somehow figure out a way to work with a little bit of lack of knowledge and a little bit of uncertainty. I thought I would also cover some broad challenges in the field of control and synthetic biology that a lot of people encounter in their work, I think. There are obstacles to building increasingly large and complicated synthetic circuits made up of various components, and some of the obstacles include imperfect modularity. So that means if you design one synthetic circuit and another synthetic circuit and you put them together, and you think they're going to still work the same, a lot of times they don't. So this is because we have imperfect modularity of our systems. Everything communicates with each other. Another thing that we have, in my opinion, is lack of robustness. So that means that we don't have, as I was telling you, all the information about all the biomolecules involved, all the biochemistry. We can't model everything to enormous detail. So this also screws up a lot of experiments in synthetic biology. So these are 2 broad challenges in the field, in my opinion. And I think that as engineers, we have an important role in coming up with solutions. For these, I think that we can develop guidelines to do better design with biomolecules. We can use modeling with uncertainty to improve our ability to predict what circuits are going to behave like. And we can use biological feedback to mitigate against some of the lack of robustness, just like we do in engineering. So hopefully with these principles in mind, you'll see them running through the actual research results that I want to show you. So here in this work, we have built 8 composed synthetic circuits, a little library of them, if you will, by starting off with the circuit on the left-hand side and continuously adding feedback loops on top of it and seeing how the dynamics of it change. We have imperfect knowledge of the biochemistry of our original circuit, and that's something that we are just going to have to account for and be uncomfortable with it as we go along. So here I'm trying to show how to modularly compose synthetic circuits when we have unmodeled dynamics and imperfect knowledge of biomolecules and biochemistry. The circuit that we are starting off with is a common type of synthetic circuit. We'll call it a pulse generator. Some of you may know it by the name of incoherent feed-forward loop. So the way a pulse generator works is that it makes a pulse of gene expression over time. And the reason why, and the motivation why we wanted to build this was because we were trying to do some targeted drug delivery inside mammalian cells. So that means that the drug would only be delivered to these cells in this area, and then as you go further out, the pulse would decay, and the rest of the cells would be off target. So into the pulse generator, we can establish similar metrics to the way that we do in engineering about its steady state error. So that means once the dynamics have finished, how far away am I from the place where I started? That could be the steady state error. And I'm also interested in the pulse amplitude so that we have a nice observable pulse in our experiments and not a really small one. So when we first tried out this circuit, what we noticed is that it had a lot of problems. Even though it's so well known in the literature and many people have implemented it, when we tried it, we found it had about 30% steady state error. It had an amplitude of 6 times amplification compared to the basal level. But we were really unhappy about the error, so that's the first thing we wanted to fix. We could just throw out the design, or we could use our engineering knowledge to just make it better. And I advocate for the second one. So here, how can we fix this pulse generator that doesn't do very well in terms of error? Well, we use our engineering knowledge, and we know from engineering that negative feedback is very powerful as a tool to force the steady state error to go very close to 0. So what we did in the circuit is that we implemented a negative feedback on top of the pulse generator that we had, and it reduced our error from 30% to about 8%. So we were pretty excited about that. And that got us thinking about how can we also mess with the amplitude of the circuit? How can we mess with both at the same time? Let's see what else we can do. So yeah, we would like to improve both the error and the amplitude. So the way that we can improve this pulse generator is using uncertainty quantification to predict the result of adding these multiple feedbacks. A lot of the time, it is our role when we work with people doing experiments to do the modeling and the theory and kind of predict what would happen so that they don't have to go through 1,000 iterations of the same experiment. So that's what we did. So here we have our pulse generator, and we're adding feedbacks on top of it. And we're keeping track of uncertainty quantification because we don't have perfect models of this pulse generator. And this allows us to make a prediction of what would happen if we added feedback to it, build those circuits experimentally, and then refine our models based on what we actually measure and see in the data. So it's a back and forth with our experimental collaborators. So far, I've only told you very abstractly about the pulse generator. But I'd like to introduce a little bit about how it works. This one is a 4-gene network. the gene names are GEM, CNTF, key, and cage. The important thing here is that the key and the cage bind together in a complex, and that's what makes the negative repressive feedback in the original circuit. So that if you induce it with inducer estradiol, then this quickly activates the cage, but in a delayed manner through the CNTF, and the key then represses the cage. And the cage is what we measure because it's tagged with yellow fluorescent protein, so we can quantify that in our experiments. So as I mentioned, this creates a pulse of gene expression over here. The interesting part about this particular implementation of the pulse generator is that it relies on designer proteins, and in particular on this de novo protein degradation switch called Degron locker. The way that Degron locker works is that if the cage and the key see each other, then the Degron is exposed, and the complex is marked by degradation by the proteasome of the cell. However, the downside of it is that this is a complicated protein-protein interaction that we don't have all the details and the measurements of. This, when we built this, this was coming literally fresh off of one of our collaborators who hadn't characterized it in vivo. And we thought about doing that. And then we also thought about what would happen if we just did our best and didn't spend our time measuring it, and instead considered it kind of a little bit uncertain and a little bit of an error that we propagate through things. And we decided to go with the second option. So here, what I initially did was to fit an ODE model to the data from the pulse generator circuit using Bayesian inference. So here we have our pulse generator as a function of time, and we have the fitting to the experimental data. The fitting is in solid lines. The experimental data is in circles. As you can see, there is definitely error. The model is definitely sort of fitting the data pretty well, but not perfectly. And the reason why is because all models are wrong. All models have flaws. They don't capture every single interaction in the actual biological system. So as you can see over here, this is created by all of the things that we don't know, and we haven't measured. Still, this model is a good basis for us to proceed, is a good enough basis for us to proceed forward. And what we can do is predict, before our experimental collaborators build this in practice, we can predict what the addition of negative feedback to this pulse generator would actually do. And it's really interesting because I guess as a control theorist, I assumed it would improve it. It would improve the steady state error in 100% of cases, but that's definitely not true. So here we have this figure with 4 quadrants. And in the top left quadrant, it improves the amplitude of the pulse, but it makes the steady state error worse. On the right-hand side, we have better amplitude, better error. Then on the bottom, worse amplitude, better error, and then worse amplitude, worse error. So in practice, our actual circuit is located in this quadrant of worse amplitude and better error, which I think is just very interesting and fascinating. I also found this quadrant in the figure that's very interesting. This stands for the feedback being broken and actually competition in the circuit over the key molecule, which there's only a finite amount of that is produced. So I thought that was really interesting to find this in part of the prediction as well. We can also play with the amplitude, and we can do that by adding something called positive feedback to it. So positive feedback just means that you positively regulate here the production of GEM. And we can make the same plot of the 4 quadrants where we evaluate whether this makes the amplitude or the error better or worse. And here we can see that we have a lot of leeway of better amplitude and worse error. So what this means is that the amplitude gets higher, but as it does that, the error also gets higher, right? I mean, that's pretty reasonable. And indeed, our experimental circuit implementation of positive feedback falls into the 77% prediction here. So now that we've made this prediction, we can actually go and build these circuits experimentally and further refine the models that we are working with. So that's what we ended up doing. So here we have another model refinement slide where basically we're refining the model by fitting it to the data of the pulse generator with negative feedback. And then the same thing for the pulse generator with positive feedback. So using these further refined models, we can actually combine them together in a pipeline, in a computation pipeline for predictability made up of 2 steps. First of all, we do validation, and then we do calibration. And then this allows us to create 8 composed systems or circuits together off of the original design. So that means we start with the pulse generator, we have negative feedback, we can have positive feedback, we can have both. And then we also created a collection of filters that I'm not going to get very much into, but they're all based on the original design. The reason why we can play around with composing these circuits so easily is because of those orthogonal proteins that are poorly characterized. We have to take the good with the bad of them. In this work, I've developed a computational pipeline for predictability, and I performed quantitative fittings to calibrate our predicted models. The summary of the talk is that we can use feedback in biology to mitigate against this lack of robustness and improve the modularity of our circuits as we compose them to each other and make them increasingly more complex. And we can also tolerate some uncertainty by doing modeling with uncertainty to improve the lack of robustness to unmodeled dynamics and all the unknown biochemistry of our circuits. And with that, I'll stop here and take any questions. Cool. Yes.
This feedback system. How many of these feedback systems have you actually made?
8 in total.
Okay.
Yeah, a library.
Over what period?
Huh?
How many years?
Pretty fast, actually. Experimentally, about 2 years. And then for the theory, I'm hiding a lot of pain that I didn't tell you about. Like the fitting, because the models are wrong, which all models are, but I explain why these ones are. It's actually quite challenging. Yeah.
And if you're going to make a new one, that's going to take another month, or?
Yeah, I think it would
maybe
be
shorter.
I think it would be pretty fast. So
Okay.
that's pretty exciting. Yeah.
Thank you. Very interesting talk. Thank you.
Yeah. Thank you. Okay, and one more question.
Sorry, just real quick, as somebody who's unfamiliar with synthetic biology, the genes that you design your circuits from, like presumably those genes were there to do like other stuff, but you figured out that, oh, this gene represses this gene, or this gene promotes that gene, so you use them in your circuits. I'm curious, like, when you build your circuits, and you want those relationships between those genes for your purposes, but when you introduce them into the organism, does it like do other stuff because each component gene was there to do something?
Yeah, so great question. So for the Degron-Locker system, which is what all these feedbacks are based on, we're pretty sure it does nothing. The other genes in the circuit that you see, they're actually taken by synthetic biologists and edited a little bit to be, some of them are edited to be inducible. So that means when you add pipette the inducer, they respond. It is possible that those have some small interaction with what's actually inside the cell. I do think that designer proteins are very exciting specifically because they don't have overlap with what's happening inside the cell. They're just completely man-made. Yeah, thank you.
Okay, hi everybody. I'm going to tell you about how we design integral feedback controllers in living cells. But first things first, so when I joined ETH about 2011, I had a shiny, brand new wet lab, and I had this dream of building an integral or PID controller in a living cell. And this was a big challenge, especially since when I joined, I was just a control theorist and I hadn't touched a test tube in my life up to that point. And so there was a long, arduous journey. And actually, it took us a good part of 7 years to get it to work. And I'll show you some of those challenges. So anyway, but first, I want to, since this is a tutorial, I want to tell you about different modalities for controlling living cells. So one way to do it is you can interface a living cell with a digital computer in a feedback fashion and use a computer to control the cell. Now, you could do it in two different ways. You can have a population of cells and then interact in a feedback fashion with a computer, or you can have a single cell interfaced with a computer. And the way we would actually interface cells with computers, you can either add a chemical inducer, or you could do, as we have done, use optogenetics and light. So light can be used as a medium to actuate the cell. And to do that, you have to engineer the cells to be light responsive. And then once you do that, then you can actuate, and of course you can use fluorescence measurements to do the sensing and use that to close the feedback loop. So I will give you one or two very, very short examples of that, but most of the talk is going to be focused on designing endogenous circuits that control the cell, particularly integral feedback controller. And here, the control system is made out of molecules, so there's biomolecular controllers using genetic circuits. So just to show you one example of the first type, so here we have a bioreactor, 1 liter bioreactor that we have built, and we outfitted it with LEDs so that we can activate the cells with light. The cells are engineered to be light responsive. And you can see here, when the cells see the light, they express a gene that codes for a protein of interest called amylase. So amylase has a lot of industrial uses. The problem with amylase is that it doesn't fold easily. So it's a difficult to fold protein. And what happens if the amylase accumulates inside the cells are stressed, and then they produce less amylase. And so we cannot measure the amylase directly, but what we have done is that we've actually hijacked the cell's endogenous circuit for sensing stress. So the so-called unfolded protein response. And by tagging this with a fluorescent reporter, we can get a feeling about how stressed the cells are. And then the idea is that to use the stress level of the cell to close the feedback loop and control a specific stress. So we'd like to stress the cells a little bit so that they can make the protein for us, but not so much that they actually have unfolded protein response and less yield. And so the bioreactor we built looks like this. And you can see various LEDs you can shine on the cell in real time. And this is what one of the runs looks like. So you can have a different set point for the stress level you'd like to see. And then the feedback controller. In this case, it's a very simple PID controller that's doing the regulation. And you could do that for different stress levels. And what we find is that if you increase the stress, the yield increases up to a point. This is a sweet spot. And then if you increase the stress beyond that, the yield actually goes down. And so you can then use the feedback regulation to lock into the optimal stress level. And in doing so, you can get as much as 60% increase in yield. Okay, so this is one example how one can use bioreactors and feedback loops in real time for enhancing yield. But today's lecture is about biomolecular control, genetically engineered circuits. And so what I would like to start with is just show you how you can actually take these genetic circuits and develop models, mathematical models that you can use to rationalize the different designs. There are two ways to do these models. You could do deterministic models or you could do stochastic models. But we'll start with a very, very simple example. Suppose you have a gene that produces a protein P. The protein can dimerize into a dimer, and it's a reversible process. The dimer, in turn, can bind to the gene, turning it off in the process. So this gives you a negative feedback loop. Now, this very simple circuit can be represented as chemical reactions, right? A set of chemical reactions. There are about 8 of them here. Each of these arrows represent one of these reactions. And the thetas are the reaction rate parameters, okay? Now, if you know these chemical reactions, the state variables, there are concentrations of these 4 species, the proteins, the dimer, the gene that's active, and the gene that's inactive. And that just evolves in the first orthont. And then, if you know the chemical reactions, you can easily write down the stoichiometry matrix, which describes how these reactions change the reactants copy number. So, for example, when this first reaction fires, you have 1 extra protein, and that's represented with this 1 here, and the other species, they're unchanged, so you have zeros, and so on. So you could do this for all 7 reactions. And then finally, the reaction rates are dictated by the reaction rate parameters and the sort of the law that you use for kinetics. In this case, I'm showing you mass action kinetics, and so that's this vector on the right. Now, if you have the stoichiometry and you have the reaction rate parameters, you can write down the dynamics of the system, and it's simply x dot equals s times lambda, so you have a nonlinear ordinary differential equation. The problem comes in is that inside the cell, you often have very low copy numbers, so you can't really represent things as continuous variables that evolve deterministically, and instead you have to represent these things as discrete random variables that evolve over an integer lattice, a non-negative integer lattice. And so here, the p's here, these are the copy numbers of the various proteins, and the stoichiometry matrix stays the same, but the dynamic model now, we have a stochastic process, which is the sum of unit Poisson processes. And essentially, what you have a description of a continuous time discrete state jump Markov process. So this is the representation. And in this case, actually, if you take the limit as the volume goes to infinity, one can show that over finite sets, this stochastic process converges to the deterministic process that I showed you before. So these are the two ways to model what's going on inside the cell. Very often you have to go to the stochastic regime because you have very low copy numbers. 1 gene, maybe a handful of mRNAs, and so on. You cannot really model these as continuous concentrations. Okay, so let's now formulate a bimolecular regulation problem. We have a network of reactions which we don't really understand fully. Maybe we have uncertainty in the parameters and the rates. It's also stochastic. Maybe there are disturbances. We would like to augment it with another set of chemical reactions, which is our controller, such that the composite system, now the closed loop, achieves some properties. So what are these properties? Of course, we want stability. By that, I mean, in the stochastic case, ergodicity. It essentially means there's a unique stationary solution to which all initial distributions converge. We would also like to get set point tracking. We would like the output of interest, XL, to be steered to a desired set point R. Right? That's our reference. And we would like this to be robust. So in spite of the constant in time disturbances, in spite of changes in the parameters, we still like the output of interest to converge to R, to our reference. Okay, so that's the formulation. Now I want to show you one feedback controller that does the job. And this feedback controller has 2 species, Z1 and Z2, and it has 4 reactions. So essentially, the first species is produced at a constant rate mu, and then it actuates the plant network. And so this is the actuator. It has another species, Z2, that is being produced at a rate that's proportional to the concentration of the variable you want to regulate. So this acts like a sensor. And now Z1 and Z2 come together one-to-one to annihilate each other. Or at least they don't have to annihilate. They could form a complex that's biologically inactive. Okay? Now, one can show that this simple motif does integral feedback, and we call this antithetic integral control architecture. And in fact, one can demonstrate that the expectation of the difference between Z1 and Z2, the concentration of Z1 and Z2, is given by the integral of this constant mu over theta, which is our reference, minus the expectation of the variable of interest. Okay? So at steady state, this expectation is a constant, and so the only way this integral will give you a constant is if the integrand goes to 0, meaning that the expectation converges to mu over theta. Notice that this doesn't depend on any of the plant parameters. It doesn't depend on any of the other controller parameters. It just depends on the ratio of those two parameters, mu and theta. And so this is on average, so the expectation. But it's also true for a single cell as a time average. Okay? So the time average of a single cell is also equal to mu over theta. And just to show you a simple example how this works, so we have here a 3-gene network in feedback with this antithetic integral controller. I'm going to start the simulation here, and at some point in time, at time 500, I will introduce a constant disturbance in the form of a degradation, and you see after a short transient, the output of interest goes back to the set point, which is 10. Then at time 1000, we just remove part of the network altogether. We remove this feedback loop and keep it off, and still the output of interest goes back to the set point. So you have perfect steady state tracking here. Now, the other variables, they assume different values, but they do so in just the right way so that the output of interest actually tracks the reference, as any good integrator will do. Okay, now it's interesting to see what kind of other controllers can do the job. And so we have, here I showed you one control system, which is an antithetic controller. Surprisingly, it turns out that if you have a stochastic model of the network, this is the only type of controller that will do it. In fact, one can show that every controller that achieves this robust perfect adaptation or steady state tracking with 0 error must embed within it an antithetic motif. What does that mean? It means any candidate controller that satisfies this requirement, if you look at the species that form that controller, they can be partitioned into 3 classes, C+, C minus, and C0. And there must be one reaction that takes one of the species in C+, combines it with another species of C minus, and with a product in C0. Okay? And so if your system is stochastic and you want to build integral controller, this is the only way to do it. Now, you can, of course, add other species which will help you adjust the dynamics, transient dynamics. As long as you have that reaction shown in red here, you will take care of the steady state tracking. But then you can also enhance the transient dynamics as well. Now, one can characterize all integral controllers, both deterministic and stochastic. I'm not going to show you this here. And so here I'm showing you 6 different simple integral feedback controllers. But it turns out that the 4 on the left, those are actually all of them deterministically will achieve the integral feedback, but only the ones on the right will do it in the stochastic regime. And they're, as you can see, they're antithetic types. The others are actually homothetic. So I don't have time to discuss this theory, but you can look up this paper and you can find out this characterization. And so it makes sense then to try to build one that works both in the stochastic regime and the deterministic regime, and this is what we have done here. So in this case, this is an implementation in E. coli. And to do that, I'm just going to describe this circuit really quickly. This here is the plant. It produces a protein of interest called RSC. RSC is sensed through this. So it activates this gene, and it produces an anti-sigma factor. So it's a protein that binds to a cognate sigma factor, which is SIGW. SIGW is produced at a constant rate. So this rate here is mu. This rate here is theta. You can adjust mu and theta by introducing chemical inducers to the solution. And so you can adjust the set point that way. And now SIGW completes the feedback loop by activating the production of RSC. So this is the simplest circuit we could think of that implements this antithetic feedback loop. And now to test the circuit, we had to build additional circuitry that act as a disturbance. So in this case, we used a protein called MF1, which is a protease. So a protease will consume the output of interest. So when you turn on the protease, and you can do so by adding a third chemical inducer, the amount of protein of interest will be expected to decrease. And that's what will happen in the open loop version of this circuit. So now I'm going to show you the experimental results. So here, first looking at the open loop, this is all in the steady state. When you introduce this protease disturbance, you lose 40% of the output at various different set points. Whereas with the circuit with the integral feedback controller, we have no measurable steady state tracking error, right? So the output before the disturbance or after the disturbance is the same. You can see this in the time domain. And so here we introduce the disturbance. And remember, this is a persistent disturbance. So it's turned on and it stays on. And so the output of interest, again, loses 40% of its value. Whereas with the integral feedback controller, there's a short transient, and then the output tracks the set point, well, as far as we can tell, without any error, at least within measurement error. So anyway, this is how we've done it in E. coli. Now, in yeast cells, and we wanted to test how these cells actually can control noisy dynamics. So we wanted to control transcription. So this time we wanted to control the mRNA. And we know transcription is extremely noisy. So to measure the amount of mRNA that's being produced, we labeled these messenger RNAs with a red fluorescent reporter. And now we also designed a system that responds to light. So when you shine light, you turn on transcription, and you can measure the output of interest by measuring the intensity of the red light. So the more mRNA, the higher the intensity. And so to see how stochastic this is, you shine a constant light on a bunch of cells that are genetically identical, and you can see different sample paths for each of those cells, even though they're genetically identical in every way, right? But you can see the effect of noise for those two cells, for example. They give you different trajectories. So in order to do this in feedback, rather than building the controller in the cell, we interfaced that cell with the computer. You can use PID or MPC, but we chose instead to simulate the antithetic controller, right, as a reaction in the cell. And of course, you have to be able to target individual cells to do that. And so we built this micromirror device that has about 4 million micromirrors that allow you to shine the light on individual cells. And so here in this setup, we call the cyber loop, the cells are in the microscope. They're constantly being segmented and tracked. And then each cell has its own private control system running in a computer whose output is fed to the digital micromirror device so that it shines the corresponding light to that particular cell. And you could do this for several hundreds or thousands of cells. So I'm going to show you a video of how this looks like. On the left, you can see the incident light on the different cells. On the right, you can see the bright spots. This is the amount of mRNA. And we had a set point of 14 molecules per cell as a target for regulation. And you can see here that the controller, the antithetic feedback controller, is tracking very well. So this here variation is due to about 100 or so cells. So if we had 10 times more cells, this would be really smooth. So you can actually really do very fine control of very noisy systems, and you don't need a lot of control signal to do it. So the controller species actually goes between 0 and 1 molecule per cell. And finally, and more recently, we have achieved this in mammalian cells, and this is where we want to go because that's where the therapeutic applications are. So in mammalian cells, we can do now proportional integral feedback using either mRNA or proteins. And so there are two different implementations. And in both cases, you can show near perfect adaptation in response to disturbances. And I want to end by showing you how one would build a PID controller. And so I showed you a proportional integral controller. Now, if you know how to make an integrator, you can get a differentiator by putting an integrator in a feedback loop with a unity feed-forward transfer function. And so this here is equivalent to S over S plus omega naught. So you have together with the integrator, you have the low-pass filter. And we've used that PID controller to control the same yeast cells that I told you before. And you can see that with the PID, you lose the peak, and the system gets a better rise time. And actually, one can show less cell-to-cell variability as a result of the differentiator. All right, I'm running out of time, I think. I will just highlight just one problem that could arise, and that is you can have dilution or cell growth that could make this a leaky integrator, and there are ways to circumvent it. And of course, saturations are expected in any time you have a gene circuit, genes saturate, and so one can devise anti-wind-up schemes for the same reason that we like to build anti-wind-up schemes for integral controllers in engineered systems. And so I will end with this. This is too much for you to see now, but since this is being recorded, you can see these references for additional work and also related work. Thank you very much for your attention, and I'm happy to answer questions.
Thank you very much, Mustafa, for your very nice talk.
Thank you.
I have a question concerning in particular, your experiments of control of individual cells. How do you deal with cell divisions? Do you restrict your control to within a generation time, or how do you go about it?
So basically, you can, once the cells divide, you can keep track of one of the two daughter cells and continue the control. And you can pick up or spawn another controller for the new cell, or you can just keep tracking one of the two original cells. Yeah. Okay.
Yes, all right. Thank you, everybody, for sticking around to the end of the first day. So my name is Harrison, or Harry, and this is actually going to be a tag team presentation with Kirill from our team. And we're here from the University of Oxford, and hopefully can tell you something about one of the areas we're working in at the intersection of control and synthetic biology, like the other speakers have all very nicely kind of set up for us. So today we're going to talk about three broad topics. The first will be about really just introducing to the idea of some of the challenges in this field around actually one of the questions that was asked earlier about what happens when you put something into a cell. Does it mess with the rest of the cell? The answer is certainly yes, and we're focusing on that question in our talk. How do you deal with those interactions between something that you put in the cell and all the stuff that's already there? I'm going to give you a specific example of such an interaction in the middle part, where we'll talk about an experiment we've done with something we built and how there was weird interactions which basically would have been impossible to work out ahead of time. And then finally, Kira will talk about a design of a system which would allow you to mitigate some other kinds of interactions of this broad family. So to start with this idea of the host circuit interactions, I guess we should think about what is actually going on in these kind of experiments that we do. So we've just seen a whole bunch of diagrams like the one on the left, where we're looking inside of a cell, we're putting together some molecules which interact with some structure or some rates that we think are useful for us. And that's fantastic, but also we have to remember that this is happening in the context of a horrendously complicated environment inside a cell, where there's thousands of other proteins and metabolites and other molecules flying around. And that's also in a broader, complex environment. And so while we might make our designs like on the left, we also sometimes get problems because of everything on the right, which is just making things more complicated and more difficult for us to predict how they will behave. And so there are a lot of different reasons for this. One of the common challenges we come across is due to this kind of idea of resource sharing within a cell. So if we imagine that I made one part of DNA here and another part of DNA on the right, we would ideally have the case that these things would just get produced by a cell and they would both behave as if the other one was not there. But in practice, I turn my DNA into some RNA, which requires some resources, transcriptional resources. I then turn my RNAs into proteins, which require some different resources, translational resources. And at both stages, they're kind of drawing from the same pool of available food or mechanisms they need to do this. And so we can see there could be some problems already in that if one guy is really greedy, the other guy might not have enough left to do what he needs to do. And this, of course, comes much worse when we then think about what's happening in the rest of the cell, in the sense that actually most of my cell is also using these resources for all of its behavior. I mean, everything the cell needs to grow and divide is being done by these resources. So if I then introduce something myself, which is taking them away, I'm going to be causing problems with the cell in general. And so these kind of ideas of resource coupling cause a lot of problems in synthetic biology and systems biology. For example, they mean that the things we create sometimes behave in unexpected ways, which I'll give an example of. They sometimes mean that because there's all this fighting for resources, cells often want to just stop doing what we're telling them to do so that they can then kind of go to a more happy state where their resources not being consumed. And this is what Kirill will focus on, how we can try and mitigate those kind of challenges with different designs. There's many more other more complex and difficult to understand challenges, but the real takeaway here is that inside a cell I have some finite amount of stuff, and if I ask it to do lots of different things at once, of course, chaos will happen because it can't do infinite things with a finite amount of resource. And so if I then realize this is the case, my next step might be to say, well, I've decided that my cell is doing something weird. How, from a control theoretic perspective, might I go about understanding that? So that then takes us to how do we actually model what's happening in these systems. And so again, in systems and synthetic biology, there's a whole spectrum of modeling complexities, from nice simple models like on the left here, where I just consider the one system I've designed and I pretend it's in isolation, and that makes my life easy, through to kind of middle complexity models where I take that system and I also somehow kind of mechanistically model its interaction with the cell's growth, through to emerging areas in whole cell models or in metabolic modeling, where actually I'm attempting to now make a model of everything happening in the cell, at least to some level of abstraction. But of course, the cell is far more complicated than we can ever realistically model in this way. So I'm still going to be making a huge amount of abstraction and assumptions to make anything tractable there. And so as biotechnologists, we have to decide somehow, where on this spectrum from something really simple that kind of ignores reality to something really complicated, which maybe accounts for various things in reality, but is also wrong, as we heard before. And somehow I have to pick a point on this spectrum that will help me understand how my system should be designed or behave. So let's look at an example of one of these weird, unexpected behaviors from a circuit we've built in our lab. And so hopefully by now we've seen quite a few of these diagrams today. This is an example of one of these biomolecular control networks where we can see at the top I have 3 inputs, my U1, U2, and U3. And down the bottom I have 2 outputs. I have Y and I have X here. And between these things, we can see there's various parts. In fact, there's 3 different RNAs, and they're doing this kind of binding with each other reaction that Mustafa has just told us about. And so in this way, you can imagine that, let's say I turn on this U3, it's going to produce some of this molecule, which will then reduce the amount of this molecule, which will then bind to, and so now I have less of this middle molecule, it's going to actually let this one on the left go up. And I can see all these different positive and negative kind of interactions between these components. And then this is something that we can actually build in a laboratory with kind of synthetic molecules that we've made, designed in theory just to target each other and implement some of these interactions. Once we've done that, we can then put it into an experimental setup. So we can put it into a bioreactor type system, which is a small device like the one Mustafa showed a picture of, where you grow the cells, you feed them, you measure. In this case, we have a red and a green output, and we can measure the red and the green fluorescence during an experiment. So I can then do some nice kind of step responses to these different inputs. So this figure here, the y-axis is the fluorescence of the system. The x-axis is time in seconds. So you can see it at this point here. I turn on the green output by turning on this vanillic acid inducer. Then at some point I turn on the middle thing, which then makes the green go down. At some later point I turn on the red input by adding this inducer, which then means the reds go up and the green also goes up because of this competition between those molecules in the middle. So I can make some very nice kind of experiments here, which kind of both qualitatively and in fact quantitatively, I can fit relatively simple ODE models to these experiments, and I can get out a model which kind of represents what I expected to happen, and I can get parameters for all these different binding reactions to some extent, in the sense that I can then predict what would happen to different sets of inputs provided at different times. But of course, this is still simplifying things. So if I look at that schematic I had before, I can see that there's these designed interactions that I've put in the middle, but there's also unexpected ones. So for example, these reporters, the red and the green colors, are fighting with each other for food in the sense that both of them are producing large amount of proteins, which the cell can't just produce infinite amounts of proteins. So I have some kind of interaction where these have a negative impact upon one another. I'll then have other unexpected interactions because actually the process of binding between these two molecules has some resources itself which need to be present for that binding to happen. And so that's now adding another layer of complexity which I don't really like to see. And then even I have, of course, all of this stuff getting done is using the production and degradation machinery of my host cell. So I can quickly see there's a lot of potential chaos here which is potentially going to cause me problems. And in fact, we do sometimes see these problems. So here's a really simple version of that circuit where I just have my red and green. And so what I would expect to happen in this circuit is if I turn on the red protein, it should go up. And so my example here, indeed, I turn on the red protein and it does go up. And then if I turned on this molecule here, which is messing with the green protein, I would expect nothing to happen to the red. Because as we can see, this arrow goes to the green protein, it does not touch the red. So in theory, nothing should happen. And yet in my experiment, things happen, which is not good, and maybe it hints that something's going wrong with some of these resources in the back end. And in fact, we can kind of dig a little bit deeper, and it turns out there are some kind of shared resources here. In particular, there's a protein called HFQ, which is kind of responsible for doing this binding between these RNAs to form complexes and then prevent them from producing things. And so we hypothesized that in this case, what was happening was maybe there was something else in the cell, so actually a native one of these molecules, which someone said, what happens if things interact? And this was exactly this case. It seems as if there was a separate molecule in the cell which just happened to be messing with the one that we'd introduced, and we could basically never have predicted this easily. I mean, we can go and do some kind of calculations about how much do we expect molecules to bind, but it turns out that this doesn't really hold the answers, at least with the level of calculations and prediction models we have at the moment. And so then we have this hypothesis. We think that there's this shared resource of HFQ. So of course, to test this hypothesis, what do? We can add other perturbations which mess with this shared resource, and we can predict what would happen. Well, we think that if I start sucking away some of this shared resource, well, now the effect of this native problem child here, the sRNA, will change. And indeed, I can make some kind of model which will see how much this should happen. And so summarizing where I am to now, I've just very briefly gone through this example, but the point has been that we start off with a model, we start off with an idea of what our system should do, and we've found problems because of these resource competitions. And the more broad takeaway from this might be that we need to have some kind of understanding of how these gene circuits behave, both within themselves, but also as they interact with the rest of the cell, potentially over long periods of time. And so for that, I'll pass to Kirill, who will give an example of how we might actually make a system which is accounting for how the gene circuits might change and behave in the host.
Yes, hello everyone, and for our in-depth example, let's consider a problem that has been mentioned in this very seminar session, that is the mutant takeover of cell populations, because when you put synthetic genes into the cells, they naturally consume some of the cell's gene expression resources. Gene expression is basically making proteins according to genes, and therefore they take away these resources from cell growth. But when these synthetic genes are mutated and are no longer expressed, then there are suddenly more resources available for the cell. So the mutant cell starts growing much faster, and eventually it will outgrow and displace the engineered cells from the population, which is, of course, a major limiting factor for the productivity and the durability of engineered biotechnologies. Now, as we have also seen, some clever design choices can allow us to tackle these problems, but coming up with creative solutions every single time is really effort-intensive. So we could really benefit from some kind of strategy that would be tunable and reusable across different applications with minimum changes. And to this end, we propose the Punisher biomolecular controller, which consists primarily of a self-activating genetic switch that is a protein which, upon chemical modulation, promotes its own synthesis. And this switch gene is also correlated with an integrase, which is a particular protein that, when its concentration is sufficient, can cut out an essential gene from cell growth from the cell's genome. And this means that without this essential gene, the cell will be unable to grow. But of course, the expression of both these proteins is heavily dependent on the availability of expression resources, that is, the burden. So when we have this Punisher circuit present in the cell alongside some other synthetic burdensome gene, first we have quite a lot of burden, so the cell doesn't grow all that fast. But when this burdensome gene is mutated, then we have more resources for growth, but also for integrase synthesis. So its level increases and cuts out this essential gene, and therefore you have a slowdown of cell growth. For example, if this essential gene is antibiotic resistance, then the cell will basically be killed by antibiotics. So this is exactly what we can see in simulated trajectories, where upon mutation the cell growth first experiences an uptick, but once the integrase catches up with it, then it plummets. Still, I have just outlined the general principle of how it should be working, but I haven't explained how, when, and why the switching will occur within the Punisher. And even if we knew that, we would still need to pick the design parameters for this kind of circuit. And predicting all that and predicting resource availability is really complicated, because besides the competition that we discussed, there's a whole bunch of other factors influencing resource pool size. That is the toxicity of synthetic proteins, the dependence of protein dilution on host cell growth, and as well, the cell actually actively regulates its own resource availability so as to maximize its growth rate in particular conditions. And if this simplified, heavily simplified diagram already looks quite complicated, you are absolutely right. But luckily, to our help can come the resource-aware coarse-grained cell models, which occupy the middle ground on the modeling complexity spectrum that Harrison has talked about. Basically, we're trying to capture this dynamic optimal resource allocation and Escherichia coli cells somewhat reliably, both in steady state and dynamically. But at the same time, we're trying to achieve sufficient simplicity to enable analytical derivations regarding the parameter dependence of the cell and the circuit. And we do that by coarse-graining the thousands of genes present in the bacterium into just a few classes based on their function and regulatory behavior. And applying this framework to our Punisher circuit, we first define the burden as an actual parameter which combines the concentrations, the promoter strengths, and the ribosome binding sequence strengths of all the synthetic genes present in the cell. And then we look at the bifurcations experienced by the Punisher. And we can see that they're actually given by the transcription rate, that is mRNA production rate for the switch gene. On one hand, we have the real transcription rate, which is defined through biochemistry with a Hill relation capturing the self-activating nature of gene regulation. But at the same time, we also have the required transcription rate, which is necessary to maintain a given switch protein concentration in a given environment with a given kind of burden present in the cell. And only when these two rates coincide, we can have a stable steady-state equilibrium. So when the burden is high enough, we will have two possible stable steady states, one of which has low expression of the integrase and the protease. But when the burden falls, for example, due to synthetic gene mutation, we're stuck with just one high expression equilibrium, which means that from this low expression to high expression of integrase, we'll have a huge upregulation of its synthesis. But of course, this kind of bifurcation threshold should be tunable, because in different applications, different genes will burden cell to different extents. And luckily, we can see by examining the real transcription rate that the shape of this curve can be changed by amending the amount of the chemical inducer present in the medium, and therefore shift the bifurcation threshold. And this is great news, because chemical inducer concentration is something that we can change quite easily by simply changing the kind of medium that the cells are grown in, and we avoid any kind of genetic re-engineering of our circuit, meaning that the same implementation, which is really costly to manufacture, can be taken from one context to another. Still, we also, as has also been mentioned, need to reject disturbances, that is, temporary decreases in burden which are not caused by mutations. Luckily, we know that in synthetic gene circuits, the dynamics of behavior are mostly regulated by the rate of degradation of the species. And in our case, we have a protease which degrades the switch protein. And by looking at the transcription rate curves, we can see that if we change the protease expression together with the inducer level, we can change the switching time without shifting the bifurcation threshold that we have so carefully tuned. Now let's consider a simple application for our circuit. For example, a toggle switch, which is a basic genetic memory device in which one gene is highly expressed, but the other is repressed. However, their fortunes can be reversed upon experiencing a temporary pulse of inducer concentration, and this is how the cell memorizes these pulses. And what's interesting is that none of these genes is guaranteed to stay highly expressed at all times, and therefore coupling cell survival to just one gene will simply not work. However, when we add the punisher to the cell, we can see that it also doesn't work when we mutate both of the toggles genes. But this just means that we need to readjust the chemical inducer concentration, and Kazam, now it does switch when the genes are mutated, but it also reacts to temporary decreases in burden when the toggle is flipped, as I discussed. But to this end, we just need to readjust the protease expression again, and now we have correct behavior for mutations and for temporary fluctuations. Still, all this is just single-cell simulations, but we're eventually interested in population-level effects. So to this end, we define a simple population model with different cells in different states, defined by which synthetic genes are present in them and which equilibrium the punisher is currently at. And we define the switching rates between these states using the simulations of stochastic and deterministic single-cell models. And when we plug all these rates in, we can see that the punisher should indeed at least double both the productivity and the functional duration of cell populations for a wide range of possible mutation rates. So we have seen how incorporating host circuit interactions into your modeling can give you a lot of insights about how a circuit behaves, but also we can see that by sensing the burden, we can actually remove the negative effects of the burden for cell populations, which is also great. But there is still, of course, a lot of work to be done in the future. We still need to develop better models and better parameterization. We need to design and experimentally implement amazing new biomolecular controllers. And if you want to learn more about it, and especially about our models that we use for analysis, you can scan this QR code over here. So I think that's it for us. So thanks a lot to our collaborators both within the group at Imperial College London, and I'll gladly take any questions.
So thank you very much for your beautiful talk. Having a Punisher circuit that is tunable is great. I mean, it's a great perspective, but nature finds all possible ways to escape what you want them to do. And I was wondering, how sensitive is your Punisher circuit itself to mutations? Can you lose it by mutations so that the whole thing fails? And if you have thought about strategies to circumvent it?
Yeah, great question. Thanks a lot. But as a matter of fact, our population model that we considered actually does account for possible mutations of the Punisher circuit. And with the right parameters, as I was saying, basically the synthetic gene mutation rates here in this diagram are defined for all synthetic genes that are present. We can still we have these kind of maintenance. And of course, if it just didn't mutate, then of course it won't be just doubling the productivity. It will mean that like infinitely we're getting rid of all the mutants. But we're not assuming that.
So today's talk is going to be about stochastic control of event timing in living cells. And I was telling Professor Astrom, I took stochastic control as my first grad course with him in Santa Barbara. So he had a role to play in at least my mathematical working on stochastic control now. So I think by now all of us know what gene expression is. So basically that's the most fundamental process in any living cell by which a protein is made from its gene. And given the complexities and the inherent stochastic nature of biochemical reactions, we have stochastic fluctuations in the level of different gene products, like mRNAs and proteins. So let me show you what gene activity looks like in a single kind of human cell. So here's a kind of a paper in PNAS from a few years ago, where you looked at activity in so-called HeLa cells. So these are kind of a cancer cell line. This dot here is a gene of interest that is on. It's making RNA. And if you recall, Each gene has 2 copies, so both copies are on, and the red is the corresponding mRNA of the gene. The neighboring cell only has 1 copy on, other copy is off. The cell over here has both copies off, but you see there's mRNA, so they must have been on in the past. So each gene is like a telegraph process. It turns on for a few hours, it makes mRNA, and then turns off. And there's some very interesting biology of chromatin loops opening and closing that lead to this kind of telegraph stochastic switching in gene states. Of course, gene is switching, and this stochastic fluctuations will propagate to the protein level, and they have tremendous consequences for biology and medicine. So here's one slide more on the consequences of noise in gene expression. So over the last 5 years, we're working closely with people in medical schools, looking at how noise in gene expression impacts drug tolerance in cancer cells, and here's some of our work in the last 5 years. So in this picture, each dot is a single melanoma cell. It's clonal, right? It's in the same environment. And most cells are blue, so they'll die upon chemotherapy. But some cells that are red and yellow have, by random chance, high levels of a single gene that makes them drug tolerant. Now, I'm showing a single gene, but we have found 100 of such genes that are all interconnected in a network. And when they are highly expressed, the cell becomes drug tolerant. So what we show is that individual cells undergo these transitions from a drug-sensitive state to being drug tolerant. They stay here for some cell cycles, and then they revert back. So we call this phenomena bet hedging. A rare population of cells is primed to respond to environmental fluctuations. And upon drug therapy, these green cells will survive. They will adapt and become resistant to chemotherapy, and that's when therapy fails. So we try to characterize this in different cancers, as you can read in this work. And basically, noise plays a very important role in helping cells respond and adapt to environmental fluctuations. Just to give you a flavor of the kind of modeling we do, so think about each gene as this telegraph process, turning off and on, and gene activity is influencing activity of some other gene. There's some complex network. The network itself is getting reprogrammed upon chemotherapy. So that's kind of the complexity here. And in this paper, we show that the right topology of the gene network can lead to these states where multiple genes turn on in a single cell and make it drug tolerant. And now we are going to these same genes, and we delete each gene, and we see if we can affect tolerance to chemotherapy. That's kind of an example of kind of work that we do in collaboration with people in medical schools and life sciences. So just to kind of sum it up, gene expression is stochastic. It creates variation between cells, which has tremendous consequences for medicine and biology. And in this talk, I'll talk about something more fundamental, is how do cells do timekeeping given this stochastic nature of gene expression. And for this, we picked an experimental model system, which is basically a phage, a lambda phage. So that's a virus that infects E. coli cells. So after infection, this cell can have 2 possible outcomes. The virus can replicate and lyse the cell, or it can become dormant, we call it lysogeny, and it can activate at some later time point. So to look at timing at a single cell level, we leverage this experimental system. And what we do is we take these dormant cells that have the phage inside them. We do a thermal induction. So we raise temperature for 5 minutes by 5 degrees, and that activates the virus. We watch this single cell in a microscope and record the time it takes to go from induction to cell lysis. And I want to acknowledge our microbiologist partners in New York University who kind of help us with all the experiments with the phages. And we do the easy part, which is the analysis, imaging, and the modeling. So what we expected was the timing to be quite stochastic between cells. There's cells of different sizes in different cell cycle stages. But amazingly, the timing was precise at a single cell level. What does it mean to be precise? Well, that's data. In these growth conditions, it takes about 1 hour for the cell to lyse, plus minus 5% fluctuations. Given the experimental error, we refer to this as somewhat precise. And now to understand using stochastic control theory, what kind of regulation and control is required to make timing so precise at a single cell level, given the stochastic nature of gene expression. So we turn to mathematical models. But unlike in control theory, where you have a black box model, in biology, you have to make more mechanistic models. We get a better understanding of the process of lysis. So to make a mechanistic model, we try to learn about how this process happens. So all phages have a protein called holin. It's a membrane protein of the virus. So once you activate the virus, this holin protein will accumulate slowly in the membrane. Of course, you can't lyse very fast, because then you don't make any progeny. So you want to really wait. So it's kind of a slow death. When this protein accumulates at a very critical level, say 1,000 molecules in the membrane, they come together to form a raft. The raft forms a hole, and the hole in the membrane leads to cell lysis. That's the process by which phages kill their bacterial host cells. So here's some movies. It's an E. coli cell infected 1 hour ago. And I'm showing the last 5 seconds. So we have students, and we make them watch these movies. They record at a single cell level timing of lysis. We have distribution. We can do it for different genotypes. We can do it for different growth rates. And then we'll have our mathematical models that I'll show you, which we try to fit to data and try to learn about the biology. OK, here's a movie where holin is tagged to GFP. So you'll see the holin diffusing in the membrane. At some point, it'll come together to form the raft for the whole formation. It's interesting, by putting GFP, the lysis time becomes double. So putting GFP actually interferes with the lysis process and makes it less efficient. So given to make a measurement, you have to have a sensor. But the sensor changes the plant, which is the kind of reality of working with these systems. OK, so now let me give you the mathematical model. And I'm going to start with the simplest model of gene expression. If you tell a biologist that I'm modeling expression as 4 chemical reactions, they may not believe it. But it's a simple system with 4 reactions, which is transcription, mRNA decay, translation, and protein decay. As Mustafa mentioned, the classical framework of this is basically you have events, which are chemical reactions. They happen at a randomly over time with some propensity or rate. And when these events happen, the population counts change by integers based on the stoichiometry of the event. So if you have a transcription event, mRNA levels will increase by 1. You can write a chemical master equation. You can do a lot of theoretical analysis. But I'll get to the main kind of theorem before we do the results. So here's basically the timing problem, right? You have expression of this gene, holin. It gets to some threshold. And then you have lysis. We call this FPT, or the first passage time. The threshold can be defined in 3 different ways. You can have a fixed threshold, a soft threshold, which means it's stochastic itself, or an adaptive threshold, where it's itself a dynamical system. So for simplicity, here is going to be a fixed hard threshold of, say, 1,000 molecules. But the time it takes to reach the threshold is going to be random because of the random process of gene expression. And that gives you a distribution of FPT, which is also what we are measuring experimentally in lambda phage. So here's kind of our main result that was published in 2 different papers. The first paper is the discrete formulation I showed you, where you have integer copy numbers. The next paper is basically looking at concentrations and taking into account cell size. So cell size itself is a dynamical variable, which we often ignore. And cell size and resource constraints are important factors that play into these kind of modeling. So we have both frameworks, discrete and continuous. And we have exact results on the FPT moments. So here, Km is my transcription rate. Capital X is my threshold. B is the burst size, number of proteins made per mRNA. And the beauty is, experimentally, I can change all 4 parameters and get my FPT. And from a model, I can predict it. So basically, observations led to our model. We are doing new experiments, which lead to new models. And that's the feedback between theory and experiments that leads to new knowledge. So here's some example where we experimentally change the threshold of lysis. And so here is different lambda phages. So the Holin protein has a sequence. So you mutate the sequence point by point and have different lambdas with different thresholds, 100 molecules, 1,000 molecules, 10,000 molecules. So these are the triangles, are the timing of lysis in these different strains with different thresholds. The y-axis is the noise, CV, standard deviation over mean, and the x-axis is the mean, time to lysis. And the black line is the prediction of the mathematical model I showed you earlier. So basically, in these timing problems, an optimal threshold is required to minimize your noise in timing. That's the basic message of this plot. The next thing is to introduce feedback control. So the basic idea of feedback control, which is the simplest idea, is that your transcription rate depends on the protein level. So in the previous case, it was constant. And now, what you can have is that, what's the optimal way to design a feedback to have the most precise timing? So let's say at time 0, you have 0 molecules, and you want to get to 1,000 molecules in 1 hour. Not half an hour, not 2 hours. So 1,000 molecules in approximately 1 hour. What is the best feedback? What is the best function Km to have that will get there in 1 hour with the lowest noise in timing? Okay, that's the thing. So I'm going to give you 4 options. No feedback, which is constant transcription. Negative feedback, which is a decreasing function. More protein, less transcription. Positive feedback, which is an increasing function. Or a mixed feedback, which is a combination of both. So given the 4 options, no feedback, negative feedback, positive feedback, mixed feedback, what do you guys think is the optimal feedback to get to a threshold in a given time with the lowest noise? Mustafa? Okay. So idea is that the surprise, in this case, feedback will make it worse. So what we showed is that for a stable protein, that's a long half-life protein, no feedback is the optimal feedback for the lowest noise in timing. And here's some simulations with negative feedback, positive feedback, and no feedback. Again, the mean time is fixed. That's the important constraint in the problem. And for an unstable protein, short half-life, positive feedback is the best feedback. This is for a stable protein, no feedback, and you need a positive feedback for an unstable protein for timing precision, not copy number, which is different. This is about timing. Now, lambda has no feedback, but it has something more interesting, which is feed forward. So what happens is that in lambda phage, the mRNA, it makes 2 versions of the same protein. So it makes something called holin, which makes the whole, but it also makes anti-holin, which essentially binds holin and prevents it from its function. So in the wild-type virus, it's 2:1. For every 2 holin, there's 1 anti-holin. Of course, if it's 1 and 1, it'll never lyse. So it has to be 2:1. And what we found is that some experiments where if you delete this anti-holin, then the noise in the timing of lysis is much higher. So now we're trying to change the ratio. We have 3:1, 3:2, 4:1, and doing some kind of experiments and both modeling to understand how this incoherent feed forward can lead to more precise timing of events at a single cell level. Okay, finally, a more slide on the ecology. We tried different lambdas with different timing of lysis. We did an experiment. We looked at the fitness of the virus. So we infect E. coli cells and look at the amount of virus after a long, long time. That's the y-axis, is the lambda titer. And this is the lysis time. As you can see, it's an optimal time to lyse. You can't lyse too early. There are not enough progeny. If you lyse too long, the host cell dies and the virus dies. And because you have an optimal time to lyse, you want to control your timing fluctuations around that optimal time, which is why you need this feed-forward control and low burst size to have timing precise. And I think this is the first experimental demonstration of an optimal time to lyse of a host by a virus that we published in MBio. Okay, just to summarize, we think noise is unavoidable at a single cell level. Noise is a double-edged sword. It can be detrimental, so it's controlled to reduce noise. Noise can be beneficial to adapt to different conditions, as we saw in the example with cancer cells. I think combining single-cell studies with stochastic models can give us more mechanistic insight that you can get with population averages. And something that's a theme that me and Mustafa, we've followed it for a while. So now I'm going to end my talk and take some questions, and I want to thank my funders, so basically NIH, NSF, and the US Army. And the phage experiments were done with John Denny and Sharon, who did all the experiments with the lambda phage. Thank you. So questions? Yes, Mustafa.
Can you give an intuition why the open loop gives you more precision?
So we think it's all about that nonlinearity.
Let me ask it again.
Yeah. So yeah.
So can you give us some intuition why the open loop gives you more precision?
So this is kind of what I think what happens, because basically with feedback, you get this nonlinear increase, right? Without feedback, it's a nice linear increase. But with negative feedback, it's the worst in timing because of this. It becomes concave. So if the threshold is a bit higher, it'll become very bad with noise. And positive feedback is slightly better than no feedback, but negative feedback turns out to be the worst if you keep the mean timing fixed. Yeah. Yes, Anya.
Yeah, so you mentioned that if the protein was unstable, then positive feedback would be the better one. What do you mean when you say the protein is unstable?
So it's all about the half-life. So
Okay.
stable protein is a long half-life, so it's mostly diluted. In this
Right.
case, no feedback is the best strategy. But if you have a transcription factor with a short half-life of, say, half an hour, minutes, it all depends on the cell cycle duration. That's the context. So if
Okay.
it's a short half-life, so it's actively degraded,
Yeah.
then positive feedback gives you a more linear response.
Yeah, and you also mentioned combining both the positive and the negative.
Oh, there was just an
There's no
example of
bottom.
option number D.
Okay.
I've never implemented it,
Okay.
but it'll be
Okay.
interesting to implement and see.
All right, thank you very much.
Yeah, it'll take a geological time scale to implement it experimentally. And given it's a membrane protein, so there's no actual way of feedbacking it. Oh, was there a question? Maybe one more question. Okay, thank you. Thank you guys for organizing the workshop.